Hello, everyone, and welcome to Indoor Biotechnologies' latest webinar on our SARS-CoV-2 proteins and simple T-cell test for COVID-19. For today, we're going to be discussing these essential tools for COVID-19 research and development. Greatly appreciate everyone taking time from their busy schedule to join us today, and we're excited at this opportunity to present this information to you. We have two exceptional speakers for you today, starting with Dr. Saya Aga, Senior Scientist here at Indoor Biotechnologies Incorporated in Charlottesville, Virginia. Saya will be speaking to our recombinant SARS-CoV-2 proteins, including a newly released full-length spike protein. Saya will be covering the manufacturing process, stringent QC measures that we put on our proteins, as well as applications. Saya will also be speaking to some interesting ELISA data that compares the nucleocapsid RNA-Binding domain versus full-length spike proteins in determining vaccine efficacy. Following Saya, we'll have Dr. Maria Oliver, Senior Scientist at Indoor Biotechnologies Limited in Cardiff, Wales. Maria will be speaking to the InBio simple T-cell test that she is spearheading development efforts on. Maria will go into detail of these development efforts, achieved milestones, as well as the future of the project. And I'm your moderator today, Beatty Sturgill, Business Development Manager at Indoor Biotechnologies Incorporated. So just to provide a little structure for the webinar today, again, we'll start with Saya and the SARS-CoV-2 proteins. Following Saya, we will have Maria going into detail on our simple T-cell test for COVID-19. And at the conclusion of the webinar, we'll have some time for Q&A. Please submit any questions using the Q&A feature. It's a button on your Zoom toolbar there. You can post questions at any time during the webinar, and we'll get through as many as possible at the end of the presentations today. And with that, I'll turn it over to Saya.
Hello, my name is Saya Aga, and today I will be introducing a suite of SARS-CoV-2 proteins that we have developed here at Indoor Biotechnologies. Next slide, please. Here we are, almost 2 years in the global pandemic. There has been more than 244 million confirmed COVID-19 cases globally, with almost 5 million deaths. In the United States alone, we have seen almost 46 million confirmed COVID-19 cases, with more than 756,000 fatalities. And we are still being daily challenged. And unfortunately, it looks like that this virus is here to stay with us for some time to come. Next slide, please. Let's take a closer look at the SARS-CoV-2 virus. I'm pretty sure you've all seen different representations of this virus with its infamous spike sticking out of it. The viral genome encodes for a multitude of proteins, 4 of which are structural proteins. These include the envelope, or E protein, the membrane, or M protein. The E and M protein together form the viral envelope. The nucleocapsid, or N protein, that binds to and protects the viral RNA. And the spike, or S protein, that binds to human angiotensin converting enzyme 2, or ACE2 receptors, located on lung human epithelial cells. And this is what that drives the infection. Today, we're going to only discuss 2 of these structural proteins, the spike and the nucleocapsid. SARS-CoV-2 spike and nucleocapsid are highly immunogenic, and most COVID-19 patients produce antibodies to these proteins. Therefore, these proteins can serve as valuable reagents in serological testing and also in research. Next slide, please. The first purified SARS-CoV-2 protein that I'll be introducing is the full-length spike. Spike is a large homotrimeric glycoprotein. It has two main domains. The S1 domain, that is the point of contact between the virus and its host through the receptor binding domain, and the S2 domain that is involved in membrane fusion and virus entry. We have generated stable hex cell lines that express the full-length spike, and we've purified it using IMAG chromatography. The InBio spike has 1195 residues, and it has a C-terminal hit tag. A sample of our purified full-length spike, as analyzed by SDS page, is displayed on this slide for your view. Next slide, please. The next purified SARS-CoV-2 protein is the spike receptor binding domain. As I mentioned, the receptor binding domain is the point of contact between the virus and its host. We have expressed this protein in E. coli and purified it by multi-step chromatography. The InBio spike receptor binding domain has 222 residues, and it has a C-terminal his tag. A sample of the purified spike receptor binding domain analyzed by SDS page is displayed on this slide for your view. Next slide, please. The next purified SARS-CoV-2 protein is the nucleocapsid RNA binding domain. The nucleocapsid, or the N protein, has two domains, the N-terminal RNA binding domain and a C-terminal dimerization domain. The RNA binding domain is the domain that is highly conserved. We have expressed this protein in E. coli and purified it by multi-step chromatography. The InBio SARS-CoV-2 nucleocapsid RNA binding domain is 128 residues, and it has a C-terminal his tag. A sample of the purified nucleocapsid RNA binding domain analyzed by SDS page is displayed on this slide. Next slide, please. Here at Indoor Biotechnologies, we take pride in our stringent quality control operations. We manufacture under ISO 9001:2015 certification, which stresses on traceability and consistency. All our purified protein products are higher than 95% pure, as assessed by SDS page and mass spectrometry. The IgG reactivity of our purified SARS-CoV-2 proteins have been determined using seropositive patient sera and in ELISA assays. The concentration of these purified proteins have been determined by 3 ways of amino acid analysis, OD280 and the CA protein assay. Next slide, please. I mentioned mass spectrometry as one of our quality control steps. We're very lucky here at Indoor Biotechnologies. We own an in-house mass spectrometry facility. Mass spectrometer is an excellent tool for protein analysis. We have several uses for the mass spec here at InBio. We use the mass spec for the quality control of our new allergen or protein lots to make sure there is lot-to-lot consistency. The mass spec helps us in the R&D stages of the new protein products that we have on hand, and we perform protein identification through peptide mapping experiments, and we also run intact mass analysis on our purified proteins. We also run customer samples for allergen and protein content analysis. To assess the purity of our SARS-CoV-2 proteins using mass spec, we digested them with trypsin and collected the data on our in-house Q-Exactive Orbitrap mass spectrometer. Next slide, please. The purity or the relative abundance of our SARS-CoV-2 spike receptor binding domain and the nucleocapsid RNA binding domain were determined to be 97% and 95% respectively. An example of a peptide mapping experiment performed on spike receptor binding domain and the nucleocapsid RNA binding domain are displayed on right. The colored rectangles, are the peptides that actually were identified by the mass spec. And when you line up these peptides with the protein sequence, you get the sequence coverage. So the sequence coverages for a spike receptor-binding domain and the nucleocapsid RNA-binding domain were determined to be 85 and 84% respectively, which is a very good sequence coverage for a purified protein. Next slide, please. The mass spec analysis of the spike, full-length spike, was a little bit different. When we first performed the mass spec analysis on the full-length spike, we only got back a handful of peptides, and this was disappointing, but we realized that the spike is fully glycosylated, so the trypsin cannot reach in and fully digest this protein. So we enzymatically deglycosylated a sample of the spike, and we reran the peptide mapping experiment. The results of this experiment is displayed in panel B. And comparing panel A, glycosylated spike, and panel B, deglycosylated spike, we see that the mass spec has been identified a large number of peptides, and we bumped up the sequence coverage from 8% to 55%. The purity of our full-length spike was determined to be almost 99% by mass spec, and the sequence coverage was determined to be 55%. A sample of the glycosylated spike along with the deglycosylated spike as analyzed by STS page are displayed on the right, on the left. If you look at the arrows, you see a migration shift for the deglycosylated spike. And it runs at its expected molecular weight of 135 kilodaltons. Next slide, please. Next, we validated our purified SARS-CoV-2 proteins for IgG reactivity in ELISA assays. In panel A, we are validating the full-length spike using 3 different serum groups and 3 different serum dilutions of 1 to 100, 1 to 1,000, and 1 to 10,000. These 3 different serum groups are internal vaccinated individuals that are displayed using green triangles. Next group are PCR verified COVID-19 patient sera, displayed by black dots. And the third group are non-COVID, non-vaccinated individuals. It is good to see that there is the first 2 groups show very high IgG reactivity against our full-length spike. And also it's good to see that the vaccinated individuals show comparable IgG reactivity to actual COVID-19 patient sera. Next. And I would like to point out to the very low background for this assay. However, this assay cannot distinguish between vaccinated individuals and individuals that actually had the COVID disease. In panel B, we are validating our purified nucleocapsid RNA-binding domain using the same exact 3 serum groups and same serum dilutions. Interesting that in this assay, the only individuals that show IgG reactivity are actually the individuals that actually caught COVID-19. Next. And I would like to again point out to the very low background of this assay. And this assay actually can distinguish between vaccinated individuals and individuals that caught COVID-19. Next slide, please. Next, we used our assay employing the full-length spike to look at the IgG titers at different stages post-vaccination. To do this, we collected internal InBio samples 1 month and 6 months post-vaccination. Then the sera were probed for IgG reactivity against the full-length spike in the ELISA assay. The vaccinated 1 month post-vaccination sera are displayed in green triangles, and 6 months post-vaccination sera are displayed with blue dots. Next, please. And we saw a significant drop in IgG levels 6 months post-vaccination. This result was both very interesting and in line with what we've been hearing on the multimedia, hence the need for a booster shot. Next. You might be wondering about this data point, the single data point in the green triangle close to 0. This individual was vaccinated by J&J vaccine, and this is 2 weeks after the vaccination. And next, please. And interesting to see that this blue dot with the arrow is the same individual 1 month post then recovering from the COVID-19 disease. So this was also very interesting to see in the assay. Next slide, please. So the applications of our purified SARS-CoV-2 proteins include in diagnostics. They can be used to assess disease prevalence in communities. They can also be used to developing rapid tests, and also they can be good tools to identify who can be a good convalescent plasma donor. And as we saw in the previous slide, they can be used for vaccine efficacy testing, and as one of our assays can distinguish between vaccine immunity versus infection. Also, these proteins can be very valuable tools in research and development of anti-SARS-CoV-2 therapeutics. They can also be used in epitope analysis experiments. Next slide, please. This is a summary of our available SARS-CoV-2 proteins. The low-tox version of the proteins is a form of the protein that has low endotoxin levels. This form of the protein is suitable for in vivo studies or cell-based assays. Usually, the lead time for this form of the protein is 2 weeks. Next slide, please. A quick note on InBio allergen and protein production capabilities. We strive for high quality, consistency, transparency, but we always have a reliable supply on hand. We are capable of expressing proteins in multiple expression systems, including E. coli, Pichia, and mammalian cells. We are fully capable of expressing and producing custom designed proteins that are higher than 95% pure, and all our protein products are validated by our in-house mass spectrometer. Next slide, please. At the end of my talk, I would like to thank the whole Allergen group, specifically Dr. Sabina Wunschman, who oversees the group, and Kathy, who has worked very hard on some of these proteins, Lisa, that finalizes all our protein lots, and Zach, who is our mass spec specialist. A special thanks to Dr. Martin Chapman, who has made all of this possible. And thanks to Dr. Anna Pomes for data analysis. With this, I would like to hand over to Maria in UK, and she is going to tell you all about her exciting new T-cell based assay.
Thank you very much, Saya, for that great presentation. So I'm Maria, I'm senior scientist here at IBL in Cardiff, and I've been overseeing the development of our simple T-cell test. Since the start of the pandemic, there's been a lot of research done investigating the immune responses to SARS-CoV-2. Initial studies revealed that virus-specific T-cell responses developed in nearly all individuals with confirmed SARS-CoV-2 infection and that these SARS-CoV-2 reactive T cells were capable of co-producing multiple cytokines, such as interferon gamma, IL-2, and TNF-alpha. From studies that have been done on closely related MERS and SARS-CoV-1 viral infections, it was predicted that these T cell responses would be enduring. And in fact, this has been shown to be true, with papers published earlier this year that have demonstrated the persistence of SARS-CoV-2 T cell responses up to at least 8 months post-infection. Next slide. So early in the pandemic, multiple commercial antibodies were rapidly developed and became commercially available. But there were some questions around their sensitivity and specificity. Additionally, there's been research articles that started to come out that reported antibody titers were declining quite quickly over time. And as Sayer's data showed, this is certainly in agreement with this. Therefore, perhaps measuring T cell responses could potentially be a better readout for population immunity. The problem is that traditional assays that are used to measure antigen-specific T cell responses, and these are things such as ELISA or flow cytometry, these require separation of the T cells or the white blood cells from the whole blood first, before you can even begin the assay. And consequently, these techniques can be labour-intensive and require specialist technical knowledge. Therefore, with funding that we got here at IBL last summer from Innovate UK, our aim was to develop a rapid, simplified test to measure virus-specific T cell responses in whole blood. Next slide, please. How the assay works is that, firstly, venous blood is drawn into a vacuum container, and this contains sodium heparin. This is an important step because it stops the blood from clotting, but it doesn't cause T cell activation, which things such as EDTA are known to do. The blood is then transported to our laboratory here in Cardiff, and we split it into 3 different tubes, 3 different stimulations. There's one tube that contains PBS only, and this is our negative control. One tube that contains PHA, and this acts as our positive control for the assay, as this causes pan T cell stimulation. And then we have our test tube, which contains the megapool of SARS-CoV-2 peptides. And this megapool consists of 470 overlapping peptides, which span the entire immunodominant regions of the SARS-CoV-2 proteome. Samples are then incubated overnight, the plasmas are collected, and then we perform the immunoassays to measure cytokine production. Next slide, please. So firstly, interferon gamma. I'm just going to show you an example of a positive and negative donor. The first graph on the left is an example of what we get when we stimulate the blood from a PCR-confirmed positive donor. So the negative sample, which is a circle shown in grey, was below our cutoff of 24 picogram per mil, as we'd expected. And our positive sample in green, as well as the test sample, the SARS-CoV-2 peptide stimulated sample in blue, are both well above that cutoff. The middle graph then is from a PCR negative individual. Again, the negative control is below the cutoff, as we'd expect. The positive sample is well above the cutoff, as we'd expect. But this time, the SARS-CoV-2 peptide stimulated sample was also below the cutoff, clearly showing the difference between positive and negative donors. So the graph on the right then is basically the summary of all 128 individuals that we recruited last year between June and December. The cohort shown in the red circles are those that have received a positive PCR test result previously. The blue cohorts are those with what we've termed suspected infection or exposure. However, they did not have PCR tests, probably due to the slower rollout of routine testing, especially at the beginning of the pandemic. And then we have the green cohort, who are individuals with no known history of exposure. In the PCR positive cohort, we can see that 97% of individuals demonstrated a positive response, along with 80% positivity rate in those with suspected infection. And this is in comparison to just 20% of donors from the cohort of unexposed individuals, who also demonstrated a much lower magnitude of response. Next slide, please. So the results from Gamma demonstrated to us that this whole blood approach was definitely a viable one. So we next investigated if there were any other cytokines that could also distinguish between T-cell responses from natural infection, as opposed to those of individuals that had not been affected. We found that interleukin-2 was actually an even better, more accurate biomarker, as demonstrated in the graph. There's a clear distinction in the magnitude of IL-2 responses between individuals with previous PCR infection, again shown in the red dots, compared to those in the green with no known history of exposure. And as you can see from the coloured box, the mean IL-2 levels rose from just 9 picogram per mil in those with no known history of exposure, all the way up to a mean of 328 picogram per mil in those with PCR-confirmed infection. And I'd just like to point out that this time, all 29 PCR individuals demonstrated a positive IL-2 response above the cutoff of 20 picogram per mil. And this is in marked contrast to just 1 out of 69 from the unexposed cohort that you can see. Next slide, please. So we also analysed multiple other cytokines, and out of 15 others that we assessed, only IL-13 and IL-6 showed any significant differences between the PCR positive and the uninfected groups. But neither were as accurate as IL-2 or even interferon gamma at differentiating between those cohorts. The magnitude of IL-13 response with most samples is very small, as you can see, it's less than 10 picogram per mil. And a lot of the results were actually below the limit of detection for the assay. And as for IL-6, despite the significant stars on the graph, there was actually no clear distinction between the infected and the uninfected individuals, as there's just far too much overlap. Next slide, please. So to analyse the results a little more, receiver operating characteristic curves were generated for both IL-2 and interferon gamma. Now, these curves provide an indication of both sensitivity and specificity of your diagnostic test. And they produce a value known as the area under the curve value, or the AUC value. And what you want is basically this number to be as close to 1 as possible. The closer that it is to 1, the more accurate your diagnostic test is. And although both AUC values for IL-2 and interferon gamma are extremely high, the AUC value of 0.9950 for IL-2 was closer to 1 than the 0.9465 value of interferon gamma. And this just really emphasizes again that IL-2 is actually the more accurate of the two as a biomarker. Next slide, please. To conclude this section, for natural infection, the rapid whole blood test can distinguish SARS-CoV-2 infected individuals from uninfected ones. And the magnitude of IL-2 and interferon gamma responses increases 36-fold and 11-fold respectively from previously uninfected individuals compared to those infected individuals. Next slide, please. So with the rollout of the global vaccination programme, we next addressed the question of if our whole blood test could detect vaccine-induced immune responses as well. So to do this, we took 2 cohorts of individuals, this time one from the UK and one from the US, and the assay setup was done in exactly the same way as before. Next slide, please. So the participants for the vaccine work were divided up into those that had no previous history of infection versus those that had confirmed PCR previous exposure. Graph A firstly shows results from the previously uninfected individuals. There was an 18-fold increase in magnitude of IL-2 responses between pre and post-vaccination. The pre-vaccination responses are shown in red, and the post-vaccination responses are in blue after the first dose and green after the second dose. we can see that following vaccination, 97% of individuals demonstrated a positive IL-2 response. Secondly, in graph B, this shows data from those individuals with a previous PCR-confirmed infection. All donors demonstrated a positive IL-2 response following the first and second doses of vaccine. But interestingly, vaccination did not boost the mean of the IL-2 response to above those seen pre-vaccination. Next slide, please. Again, we analysed multiple cytokines in case we missed any other important ones, and actually only 3 others showed significant differences again. As expected, we did see great response to interferon gamma again. Mean interferon gamma levels, which are shown in graph A, increased by approximately 10-fold following vaccination. Although, as with the natural infection graph, the distinction between the groups isn't quite as clear-cut with IL-2. With IL-13 shown in graph B, the magnitude, as with natural infection, was again very small. And there were some vaccinated donors who did not produce a positive IL-13 response at all. And with IL-10, the magnitude, again here, very small. And although there was a significant difference between the pre-vaccinated samples and samples taken after the second vaccine dose only, as with interferon gamma and IL-13, there was again quite a lot of overlap between the cohorts. So we can't really say again that IL-13 and IL-10 are particularly good biomarkers. Next slide, please. Okay, to conclude the work from the vaccine-induced responses, the mean IL-2 response of previously uninfected individuals increased by 18-fold after vaccination, with 97% of those individuals demonstrating a positive vaccine-induced IL-2 response. Consequently, our conclusion is that IL-2 is a highly accurate biomarker for distinguishing unvaccinated and vaccine-induced T-cell responses in our whole blood test. Next slide, please. For my final couple of slides, I just want to discuss the next steps for the development of this test. Although our results are extremely promising, the test is not yet fully validated or ready for commercialization. However, I'm really pleased to announce that we've just been awarded a new Small Business Research Initiative grant from Innovate UK to further develop it. So over the next 12 months, we have 4 major milestones which we wish to achieve through this grant. The first 2 are being focused mostly on method validation and development, and I won't go into those here. The third milestone, however, is really where the new innovation lies in this test, and this is going to involve establishing a minimum volume of blood that we can still yield a measurable amount of cytokine from. And the reason for this is if we can get down low enough, the aim is to compare responses from venous blood draw with responses from capillary blood draw, with the main goal of this being so that we can make this test a far more accessible one, as it would no longer require the need for a trained phlebotomist to be present to take venous blood. Next slide, please. So milestone 4 is going to be done in conjunction with some amazing collaborators that we have from the NHS South Tees Hospital Trust. And with their help, we're going to put this test into a clinical setting where we hope to address the critical question of if there are differences in magnitude and longevity of vaccine-induced T-cell responses in clinically vulnerable individuals, or those individuals that are deemed more at risk from infection than immunocompetent individuals from the wider population. This work will be monitored by the immunity arm of the National Core Studies programme, which has been set up by the UK government. And we really hope that the information gathered by us over the next year will help aid decision-making on public health policies and vaccine efficacy. Next slide, please. So that just leaves me to acknowledge all the fantastic people who have helped with this work, specifically everybody here at IBL in Cardiff, who are absolutely fantastic teams to work with. Then we have to thank Dr. Martin Chapman, Dr. Nicole Brackett and Brian Smith from Indoor Biotechnologies Inc. in Charlottesville. Dr. Hannah Hoff, who did a tremendous job in taking blood from multiple donors for both the natural infection work and the vaccine-induced work. And then Dr. David Chadwick and team, who are our amazing collaborators from the South East Hospitals NHS Foundation Trust. And also then a massive thank you to Innovate UK and SBRI, who have funded this work and funding the work to be done in the next 12 months. With that, I will hand back to Beatty.
Great, thanks Maria, as well as Saya for those excellent presentations. All of these technologies and products culminate into Indoor Biotechnologies' expertise in action, in which we use our proven scientific acumen to help find the best solutions for our clients. That includes our purified proteins. Saya spoke to our available suite of recombinant SARS-CoV-2 proteins, but we also have a vast portfolio of purified allergens and rhinovirus proteins. As Saya mentioned, these proteins are purified under an ISO 9001 certified quality management system, validated via mass spec. We have various expression systems, natural and recombinant, as well as a low endotoxin product line and a biotinylated product line. We also, beyond these stock proteins, offer custom purification projects based on clients' needs. Beyond our proteins, we also have a deep portfolio of immunoassays. One product line is our ELISA 2.0 kits. These are pre-coated plates that come with all the necessary reagents to have results in just 2 hours, as well as our Maria technology, which is our multiplexing array that can detect and quantify multiple antigens from a single sample. Now, these immunoassays are available as kits, but also we offer this as a testing service to clients to send their samples into our lab, and we can do the analysis both in Charlottesville, Virginia, as well as in Cardiff. Just like the proteins, we offer custom immunoassay development as well. Indoor Biotechnologies also has viral testing capabilities. Maria spoke to our expertise in viral immunology. We use that in a new state-of-the-art facility that operates at a biosafety level 2 and is ISO 17025 compliant. And this testing capabilities can aid clients in establishing viral efficacy claims on their products. We also have an environmental testing suite in which we are able to test the safety and efficacy of products and procedures for allergen, denaturing, removal, as well as barrier testing. Saya spoke to our mass spectrometry suite that we use to validate our proteins in-house. We also offer those as services to our clients. These services, standard services, include protein identification, intact mass, as well as protein profiling of complex samples. And this can, again, as I mentioned, be a very powerful tool in characterizing extracts or samples. So all these products and services help our clients achieve what we like to call research made real, where clients are able to speak directly with InBio scientists regarding their research and product development and validation efforts, and to help realize those goals. So with that, we'll conclude the presentation portion of our webinar and move into the Q&A section. Again, please submit any questions using that Q&A feature on the toolbar. I'll stop my screen share here and pull up the questions. So we're getting some several questions here. I think we'll just get this one right off the bat here regarding distinguishing between different types of vaccines in both assays. Saya, I know you spoke to that a little bit. Maybe if you wouldn't mind providing a recap first, and then Maria, we can go to you. Saya, you're muted.
Yes, if you could please pull up one of the slides that I was, the IgG titer analysis on the post-vaccination slide. Yes, this. Thank you. So this also obviously it needs more research and more samples running on ELISA and doing the assay, but so far the majority of these samples, these individuals received the Pfizer vaccine. There are 3 individuals that received the J&J, and they are the lowest antibody responders. I have had 1 individual with the Moderna vaccine, and that individual has the highest IgG titer 6 months post-vaccination. So again, this is in line what we have been hearing. But obviously it needs more samples to be able to make a comment for sure. But this is what I have been seeing. So.
Sure, great. Maria, I don't know if you're able to address that question as well.
Yeah, absolutely. So for both the vaccines that the majority of people received in the UK cohort over here, it's between the Pfizer and BioNTech vaccine and the Oxford AstraZeneca one. And I've done some correlation analysis on the IL-2 magnitude between these 2 vaccines. And interestingly, we didn't actually see a significant difference between either of them. Now, obviously, it is standardized to some extent because we're using 1 ml of blood, but there's a lack of standardization purely because we don't do a T cell count before we actually run the analysis. So we can't say for sure that they are the same. Perhaps if we quantified them in terms of actual numbers of T cells, which is something we're actually want to, excuse me, we'd like to do with the next grant, we'll be doing that. It seems at the moment there is no difference between the vaccines. It seems that both are inducing great T cell responses.
Great, thank you. Here is another question that I believe will be to both assays. What is the smallest volume of blood you need to perform the assay? I believe this may have been directed towards Maria, but Saya, I think it would be helpful if you wanted to mention that as well.
Of course. The lowest volume I have tested was 2 to 3 microliters, and that's more than enough to perform the assay for the IgG titer analysis.
So for the T cell test, obviously we've been using 1 ml of blood, and from that we can generate about 300 microliters of plasma. Obviously, we're going to go for the next grant. We're going to see how low we can go for that. That's more than enough for the tests we're doing. And interestingly, it does point towards, again, what we would rather use IL-2 as the readout than interferon gamma, because currently interferon gamma is an ELISA kit, which requires between 50 to 100 microliters of plasma, whereas the IL-2 is done by multiplex array, and you need just a tiny amount of volume of plasma for that. So the samples are diluted 1 in 4 on that assay, and you only need 25 microliters of that. So you can see that the volumes for IL-2 are just approximately 5 to 10 microliters of plasma, so it's very small.
Great, thank you. Saya, I have a question here for you. Why is the RBD, so receptor binding domain, made in E. coli rather than hex cells?
Yes, so the main reason is feasibility and the ease of expressing proteins in E. coli versus the hex cells. Obviously, hex cells are the better expression system, but generating stable cell lines to express these proteins takes 2 to 3 months minimum. And receptor binding domain is a small protein, is around 23 to 23 kilodaltons, and it was feasible to make the protein in E. coli versus the spike. It's absolutely impossible to make the spike in E. coli, so that's why.
Okay, great. Yeah, no, that sounds great. Another question for you, Saya. With the SARS-CoV-2 proteins, Dr. Ga mentioned 3 methods for measuring the proteins. Is any method preferred, and do you get good agreement between those methods?
Yes, so the preferred method is amino acid analysis, and it's to my knowledge, is the most accurate method of protein concentration determination. We always, but it's obviously not feasible every time that we are working with proteins in the lab. We do OD280 and BCA assays side by side, and most often the amino acid analysis agrees really well with one of the two other methods, either agrees with OD280 or BCA assay, or all three. In the best case scenario, all three agree. So that's how we pick the better method in the lab to determine the protein concentration, be it OD280 or BCA. But the reference is always amino acid analysis.
Great. Saya, again, I believe this question for you. Did you test patients, Sarah, that had been infected by other coronaviruses with both spike and RNA binding domain?
I have to say, yes, I did. When I was determining the background for my assays, I tested sera and plasma that we had in the freezer at -80 from post-COVID time. And I tested a large number of those sera against both of these two proteins. And I did not see much activity, IgG reactivity to the full-length spike, but interestingly, there was some background IgG reactivity to spike receptor-binding domain. So, and those are individuals that are caught common cold, that some of common cold viruses are coronaviruses also. So,
sure.
Yes.
Great, thank you. Let's see, Maria, what is the stability and shipping conditions for the heparinated blood samples?
Okay, yeah, so we've been getting the blood to the lab within 24 hours of blood draw. They just need to be kept at room temperature and are shipped to us just at room temperature. I've tested the stability up to 30 hours post blood draw so far, and I've gone down as low as just a few hours post blood draw, comparing them over time. And the good news is we don't see any significant drop-off in responses between, say, 6 hours versus 30 hours. I haven't extended that yet, but it is something to do on the next grant to see if we can go out longer. But generally, if the samples are coming from within the UK, they can get to the lab within 24 hours. Same for the US, if they're done within the same day and get to the US lab, they're processed on the same day or on the next day at the earliest.
Great. Yeah, and again, just to reiterate that I'm assuming that'll be part of milestones 1 and 2 of the method validation.
That's right, yeah.
Great. Maria, which vaccines were received by the interleukin-2 response experiments?
Yes, so the vaccines here were the Oxford AstraZeneca one and the Pfizer BioNTech ones. We did have one individual who received the J&J vaccine, but we didn't have those second time points for that one. So yeah, the majority of them were those two main vaccines. We haven't tested anybody yet with the Moderna, where we'll just see what we can get with the next grant, see who's been given those.
Oh, great, thank you. Maria, another one. Have you done an assay in samples from unvaccinated who are post-COVID infection to see T-cell response in comparison to vaccinated?
Unvaccinated post-COVID? No, is the answer, purely because it's actually quite, especially in the UK, the rollout of the vaccination program was so quick that actually it became very difficult to find unvaccinated donors, let alone unvaccinated post-COVID ones. So no, haven't done that, purely because those people are actually really hard to find over here now.
It becomes a bit of a tongue twister saying vaccinated that many times in a sentence. Saya, back to you. can one remove the his tag from your purified SARS-CoV-2 proteins?
I have to say that is a yes for our full-length spike, because there is a proteolytic cleavage site designed right before the his tag. So that is a yes for the spike and is a no for the other two proteins, not for receptor-binding domain nor for the RNA nucleocapsid RNA-binding domain. So.
Thank you. And how about, will a low endotoxin form of the spike, assuming the full-length spike, be available?
That again is going to be available on demand. 2 weeks lead time for our current stock, depending on the amount being ordered. If it's the amount is higher than what we have in stock, it'll be probably 2 months.
Sure. The important part is that we are able to get the endotoxin removed from the full-length spike.
Yes, it actually, as its current status is not high endotoxin level, the lot is kind of lower, but we can make it even lower than that by prepping further.
Thank you. I've got a comment here. This great presentations. This question is from Maria. You measure some T cell responses in pre-vaccinated, non-infected individuals. Do you think these are cross-reactive spike-specific responses from other coronavirus infections? If so, could you exclude peptides from the stimulation that are highly conserved amongst other beta coronaviruses?
That's a good question. So I have actually done some stimulations with a couple of other common cold coronaviruses. I didn't present that data here because it's actually quite in-depth, quite time-consuming to explain it. I have looked at peptides from OC43 and 229E. They are common coronavirus viruses that cause common colds. What I saw is that some people that responded to both of those, who obviously have been infected relatively recently with those viruses, not all of them gave a SARS-CoV-2 response. Some people that were positive for SARS-CoV-2 via PCR did give responses to both or one or the other of those as well. So because we're not seeing in every PCR infected individual a response in those two other viruses, I'd say it's unlikely. I think if they were cross-reactive to the spike-specific responses, we'd see a positive result in all of them. We can't rule it out. You could potentially exclude those spike-specific responses from the peptides, but I think you then potentially run the risk of then getting a false negative if you took those out. The whole question of cross-reactivity is an extremely difficult one, and it is something that we are going to try and explore again in the new grant. But yeah, excellent question. Yeah,
great. Yeah, thank you for that question. Maria, another question. How did you determine the threshold limit for the cytokine response?
Yeah, so this is done based on the graphs I showed with the receiver operating characteristic curves. From those, you can also do further statistical analysis, and what you can generate is something called a UDENS index. UDENS index basically is a statistical analysis to give you a readout for specificity and sensitivity in both one go. And what you're looking for from a diagnostic test in general is the value that gives you the highest readout for specificity and sensitivity combined. So basically, UDENS index can measure both of those readouts at specific cytokine levels. And where that one is, the result that is the highest will give you the threshold that you should be using for your test. So it's the statistical analysis is how you determine the threshold.
Great. Another Maria. Is there a correlation between magnitude of cytokines produced and length of time since infection or length of time since vaccination?
That's a great question. Obviously, that is ultimately, that's something that we're going to be addressing in in the next grant, is the T-cell response waning, and are we already seeing that from what we've done? What I can say is that so far, for the natural infection, obviously, again, these were the unvaccinated ones. I've actually just noted a couple of numbers down here, if I can give them to you. So we took, people were tested anywhere between 2 weeks post-infection all the way up to 38 weeks post-infection. And the results for that, the person that was only infected 2 weeks prior to their test, they gave us a result of 98 picogram per mil. However, the person that was infected 38 weeks before the test actually gave a result of 770 picogram per mil, so 7 times higher. So from that alone, you could say that no, it doesn't look like the T-cell response is waning, at least 9 months later. There may be a slight trend, but it's looking quite hopeful that the T-cell response is going to be more enduring than perhaps the antibody response. And obviously, again, the next 12 months are going to be really useful for finding this out.
Mm-hmm. Yeah, great. I think Sai's presentation, that data was clear that the antibody response is waning over time. Let's see. Maria, is there a correlation between magnitude of T-cell response and antibody titer?
So we haven't actually looked at that in this set of people. Again, it's something in milestone 1 and 2 we're going to be doing, and we're going to be doing that in collaboration with some of the academics here. So I can't say for sure from the data we've got, but what we have got was when these people donated blood, we asked them to fill in a questionnaire, and that gave us information about when they were PCR positive. But they also, a lot of these people were frontline workers, so were routinely tested by lateral flow for antibodies. And so they also gave us their lateral flow results, and we had 2 or 3 people that were PCR positive, and at the same time as their PCR test were antibody positive. And yet over time, they then started to give a lateral flow antibody negative result. So at the time the test was done, they were testing negative for antibodies, but they were testing positive for T cells, which suggests again that maybe there may be a correlation in some people that produce a response to both. But again, it seems likely that maybe with the waning antibody response, whereas the T cell response may be more enduring, we would expect perhaps there to not be that correlation there.
Great. How about, were there any positive cytokine responses in the negative control simulations? Or stimulation, excuse me.
Yeah, so for the negative stimulations, obviously that to make sure that we're not just seeing unspecific cytokine stimulation. For interferon gamma, we do actually see sometimes low-level background stimulation. The cells are just ticking over and producing low levels of interferon gamma. And from those, we do actually take that background level off of the test result. But again, for IL-2, we do not see any positives in the negative control, which is great news. And again, perhaps another indication of the IL-2 assay being slightly better than the interferon gamma one.
Great. Well, I believe that's all the time we have for today. Sai and Maria, again, thank you very much for those presentations. It's clear some very important research is going on here. So thanks for sharing that. I'd like to pull up a quick slide here just to thank everyone for, sorry, let me scroll to the bottom here. Thank everyone for attending the webinar today. We will be sending out a webinar recording to all those that have registered in the following weeks. But in the meantime, if you have any questions or comments, please feel free to email myself at bsturgill@inbio.com or Maria or Saya. They'd be happy to address any questions. You can also catch up on our past webinars on our YouTube channel there, including a webinar on the allergen variability in early introduction foods, one on the molecular approach to allergy diagnostics, and one on the environmental testing suite and our viral testing capabilities that I spoke to briefly earlier. But again, appreciate everyone for attending. We always enjoy presenting this information to you, and we'll see you on the next webinar. Bye.
Bye.